So this is a CBC machine, and under the menu button, we prime the system every morning, and then we run controls on this machine weekly for it. And then every couple months we have to do a complete quality control of it to reset the system so that we know if there are highs and lows within what shows up on the screen. We know that it is not faulty and that what is showing is accurate. And when cleaning this machine, we prime the system, and it would run a cleaner through it and into here, and so it flushes out the system, and we do that every morning so that the system is clean for the daily use. When performing an ELSA test, you want to use 1 drop of blood in this sample over here, and then you want to use 2 drops of the buffer, and then you'll need to wait 10 minutes to show that if the test is accurate or not. Otherwise, you could get a false positive if you wait too long, or you may think it's negative because you haven't waited long enough. When doing a fecal float, you want to make sure that you have the fecal lube, and you want to have it lubed up so that it slides in gently into the rectum. You want to make sure that you have an adequate amount. So solution, which has salt in it to make anything in the fecal rise up, such as the intestinal parasite, eggs, or any other bacteria or giardia or anything. So you want to make sure all of it comes loose in the fecal solution, and then fill it up to the top, and then set a slide over it and let it sit for 10 to 15 minutes so that anything in there can rise up to the top and it'll be on your slide. After the fecal has sat for 10 minutes, you want to put a cover slip slide on it, and then read it under the magnification of 10, and you will be able to see any other roundworms or hookworms or whipworms that you will see. You will not see any tapeworms, or on the rare occasion you'll see the baskets that they are kept in. When getting a fecal for a fecal smear or a direct smear, sometimes you don't get enough fecal matter. Once you've collected the fecal, you want to make sure that you apply the fecal solution about as much as fecal matter as you have. Okay, and then once the fecal and solution are on the slide together, you can get a wooden applicator and mix them together till they're mixed together very well. Then you want to smear the solution across the slide and remove any large excess pieces. And then you'll want to place the cover slip over the thin amount on the slide, and then you'll be able to read it on magnification of 10 or 40. This is our dry chem machine. This is where we run the panels such as total protein and glucose and BUN and total bilirubin. And so when loading the slides, I picked a pre-surgical panel, and so it's a mini panel. So you would load the slides, make sure they're all facing the same way, and loading it as the picture describes. And then you want to set the weight on top of it. And then when running, you want to make sure that you have all the tips filled in the appropriate ones. So I'll be running the A and B. I won't be running electrolytes. And when using this piece, the sample I have is 1.5 mils, so I want to make sure that I match it with the 1.5 on here. And I'm going to use the blood sample and do the top and get rid of any fiber that is left on top. And then I'm going to stick it where it says to put the sample, and then do the ID of the patient that we're running, And then make sure it's on the right animal. So this is on a dog, and you would hit start to run the sample. Whenever the sample is done running, it will also give us a paper copy printed out on top, and it also electronically will send over the test results to our software. Ear swab, you want to make sure that you haven't already flushed out the ear. So you want to go down deep enough to make sure that you get the specimen that you want if there's anything growing within the ear. Alright, when preparing a slide for an ear cytology, you want to make sure that you firmly press it across the slide. And that you have it enough to where that you will be able to see any organisms that may be presented in the ear. Once the slide has been heat fixed, then you want to stain it, but you don't want to dip it in because the specimen is not sterile. And so you want to do a coat of the dye over the top of it. And you'll want to let it sit there for about 10 minutes. And once the slide has sat for 10 minutes, you can slowly rinse off the stain. And then you'll want to heat fix it again. When looking at specimen for an ear cytology, you're looking for like cocci and yeast and bacteria, which are very microscopic. So you want to do it on the 40 plus on the lens. And this dog has presented with yeast and chronic yeast problems. And so they will be treating it as followed. When doing a necropsy, you want to make sure that you wear a gown, gloves, and a mask so you can protect yourself from the animal. But unless you're doing a culture on the animal, then you want to make sure that you don't contaminate the sample by touching it yourself.
Come. I'm just looking for any abnormalities, looking for anything that may have resulted in the death of this animal. Externally, the only thing I'm seeing is a very thin kitten.
You need the light on already.
Yeah, that's just turning around. I'm just making a midline incision on the cat, going pretty much from stem to stern. So the entire length of the body. No reason for shaving this animal.
It's got urine.
And again, I'm just opening it so I can reveal the organs on the inside. And cut through the sternum. So again, now I'm just looking for things that are abnormal. And they look normal. This is the heart. And it looks normal size for this kitten. And then the lungs for the opposite side are here. This is the diaphragm. Separates the lung space from the abdomen. This is the liver, and there are multiple lobes to the liver. And then this is the gallbladder, which resides inside the liver. We have a stomach, fairly large stomach. We have the intestines. Very small piece of pancreas there. The intestines just go down until we get to the large intestine. And off the end of the large intestine, where the large intestine and the small intestine come together, we have the cecum, and at the end of the cecum would be the appendix. Here's a kidney. And a kidney on the opposite side. So we're going to go ahead and take some samples.
So I'm filling one of the sample bottles with the formalin for when we take a sample of the animal. You want to make sure that you always wear gloves when using it.
Okay, we're going to take a sample of a lung lobe. We're going to take a sample of the liver.
Do you need to keep them separate?
No.
No?
No. We're going to send them all to the lab at the same time.
Okay.
And we're going to take a sample of the kidney. It's important that you don't overfill your sample container, so you have enough formalin to act on all the tissue that you're putting in there.
And so I'm going to be labeling this bottle with the patient's name and what species and how old they were whenever they passed away. And then we'll submit this sample to the lab. Precautions need to be taken when handling a suspected rabies. So the people that can handle it would be the ones that are trained to handle it, such as people that work at the shelter or veterinarian, because they will need to be quarantined for 10 days at least to see if they have bitten a human or bitten something else, that they want to make sure that if it's still alive at the end or if it is showing any symptoms of having rabies later on during those 10 days. And protective gear that will need to be worn is you don't want to have the saliva of the animal near you, so you want to make sure that you are completely covered up so when handling the animal there's no chance of getting the saliva on, so you want to make sure you have no skin showing to where there's a possible bite or a possible sign of the saliva getting into your blood system. When submitting a sample, you want to make sure that you're submitting the brain tissue of the animal that is suspected of rabies, and you'll want to ship it in a cooler box that keeps it cool while being shipped so that it can stay at a cooler temperature, so and with ice packs with it as well, and you want to make sure you ship it quickly. What to do with the remainder of the body if an animal is suspected with rabies? You want to make sure that you don't get any of the blood, any of the saliva or feces on you. So you want to make sure that you double bag it and then either burn it or you can bury it. Just make sure that no animal could ever come in contact with it or they have a chance of being susceptible to the rabies. When doing a vaginal cytology, you want to make sure that you start by washing off the vulva and getting any debris from the outer side of it, because that can cause contamination when doing the swab. You want to make sure that the swab is sterile so you don't get any contaminants when doing it or any other cells on it. So I'm doing this, and then you want to make sure that it's moistened. And then you start by inserting it up dorsally. And then you want to go posterior and make sure you're over the pelvic rim. When preparing the swab, you want to press it firmly against the microscope slide and rub it across it. And then you want to heat fix it. And then when applying it to the stain, you want to do 10 in A. And then 15 in B. And 20 in C. And you can rinse it off with water. And then you want to heat fix it again. Based on what the slide presents, the dog is in anestrus, which is the beginning stages of the estrus cycle, and that one can last up to 4 to 5 months. And I found non-quantified squamous epithelial cells, which would show that she is not in estrus. Anestrus is the first cycle of the estrus in animals, and so you will see non-quantified squamous epithelial cells. You may see some neutrophils, but you will not see erythrocytes while the animal is in anestrus. And the epithelial cells in proestrus, which is the second one, where the vulva is starting to swell and a little reddish discharges, but they are not being attracted to the males. But the erythrocytes will start to decrease and the epithelial cells will start to show cornification. And estrus is the third stage, and that is when they ovulate. And whenever the epithelial cells are fully cornified and the vulva is swollen and they are starting to attract males. Metaestrus is the last stage, and that one can last 2 to 3 months, and that is when the cornified epithelial cells are starting to transform back into the non-cornified squamous epithelial cells on the slides. Okay, so when applying the slide and the blade for skin scraping, you want to apply a little bit of the mineral oil here and also on the blade you are using. Okay. So I'm going to apply some propylene glycol to the area, and I'm searching for mites, so I'm going to pinch the skin to try to see if they'll come up. And then I'm going to scrape the area, and you want to make sure when you scrape the area, you scrape it hard enough to where you get blood. So I'm looking for mites, either Demodex or Sarcoptic. The animal was presented with a patch of alopecia on the skin, and so that is what I'm looking for, and those would be the possible results that I would find. I'm pulling the blood off, or the serum off of the whole blood. Off the 2 samples. I'm pulling the plasma off of the blood samples and the EDTA tubes from the recipient and the donor. So from the animal to be tested, I'm going to pull 1 to 2 drops of blood in a glass test tube. And then I'm going to fill the rest up with 0.9% sodium chloride. You want to gently mix it together, and then you want to centrifuge it for 1 minute. So once it has centrifuged for 1 minute, you want to remove the whole top layer during the wash. And then you want to repeat this 2 more times. And then you'll centrifuge it for 1 more minute. Okay, and now I'm removing the top layer again. And then adding the sodium chloride, and filling the rest of the tube up, and centrifuging it for another minute. Now I'm removing the top layer again. Okay. Now I'm pulling 0.02 mils of the packed washed erythrocytes. And placing it in a test tube. Okay, and now I'm applying 0.98 mils of saline to the test tube along with the wash packed erythrocytes. Okay, so now I'm going to do 2 drops of the donor serum in a brand new test tube. And then 2 drops of the donor cell suspension with the donor's test tube. And then we'll gently mix the solution together.
3.
And then to make the minor cross match, we'll take 2 drops of the donor cell suspension, or the serum from the donor. And then we'll take 2 of the erythrocytes suspension and apply 2 drops there. And then when making controls for one of them, for the donor, you'll want to take 2 drops of the erythrocyte suspension mixed with its own serum. And then for the recipients, you want to do 2 drops in 1 tube, and then you want to, with its own cells, and you want to mix it with their serum. And then you want to place all the samples on a rack to have them mix gently. And then once they've mixed gently after a few times, you want to have them incubated at room temperature for 30 minutes. And then after the samples have been sitting for 30 minutes to get to room temperature, you want to centrifuge them for 1 minute. Once the test tubes come out of the centrifuge after being there for 1 minute, you want to tap the glass to see if there's any agglutination in them that you can visibly tell without using the microscope. If there is no grossly visible agglutination, you want to take a small drop, place a small amount on a glass slide, and check it under low power mode on the microscope to check for agglutination. And then you want to check the other one as well. And based off the interpretation on the slide, no hemolysis has been detected, so that these two animals would be a good cross match.